talk about in 3.6 is enzyme cofactors. And so cofactors are just non-protein molecules kind of assisting that enzyme to do its function. These cofactors can be inorganic, they can consist of metals, they can bind really loosely or tightly. A holoenzyme is just an enzyme with a cofactor attached. Okay, coenzymes have organic cofactors like vitamins. All right, and so then we also have enzyme inhibitors. And so enzyme inhibitors are going to decrease the activity of specific enzymes. They're going to target a specific enzyme to allow its function to decrease or halt in completion. And so this can be permanent or it can be reversible. If it's permanent, the inhibitor is binding with strong covalent bonds. This can be like a toxin or a protein. If it's reversible, the inhibitor itself is interacting weakly, meaning it can be removed from the enzyme. And so enzyme inhibitors, there's a couple different types. So a competitive inhibitor reduces enzyme activity by blocking substrates from binding to the active site. And so this inhibition can be reversed with increased substrate concentrations. Okay. And so here is our normal substrate binding. Okay, our substrate binds to the active site of the enzyme to allow it to do its job. Okay, but a competitive inhibitor will bind to a portion of that active site, and it's similar in shape to that substrate, but it's not the same thing. And so when it's missing a portion of the substrate, it inhibits the reaction rate. It decreases its ability to do work. And so this is competitive inhibition, when it looks like the substrate, but the increase of substrates concentrations can reverse this. And so another form of inhibitors would be the non-competitive inhibitors. And so these inhibitors are going to bind to an area other than the active site, or the allosteric site is what they call it. And so this is going to change the shape of the active site when a non-competitive inhibitor is bound to an enzyme. Okay, so we have normal substrate binding occurring right here, right? Our substrate binds to our active site. Our enzyme can produce and do work. Okay, but when a non-competitive inhibitor binds to another location other than the active site, allosteric site, it changes the shape of our active site and prevents substrates from binding. Okay, see how it changed our shape of our enzyme? And so now this shaped substrate will not fit in this enzyme, and it cannot perform its job. And so when you change the shape of that enzyme, it's pretty permanent. Now we're going to talk about the regulation of chemical reactions. And so a cell must be able to regulate metabolic pathways. Whether we don't always need a lot of different proteins in our bodies, but when we do need it, we need to be able to make it and make it quickly. And so a cell is going to regulate what goes in and what gets made, etc. And so we're going to control where and when enzymes are going to be active. And we're going to switch genes that code for those enzymes on or off. And we'll get into the details of turning genes on and off in Unit 6. Okay, and so allosteric regulation, or active site regulation. allosteric enzymes have two binding sites. Okay, we have that inhibitor binding site or the allosteric site, and then we have the active site. Okay, and so that active site is where the substrate goes. The allosteric side is the regulatory site. That's where the regulatory enzyme or regulatory substrates are bound. And so allosteric regulation, molecules are going to bind to the allosteric site, and it's going to change shape or function of the active site. So this might result in inhibition or stimulation by an activator of enzymes. And so an allosteric activator binds to the allosteric site and stabilizes the shape of the enzyme so the active sites remain open. And so it kind of goes against the inhibitors. An allosteric inhibitor substrate binds to the allosteric site and stabilizes the enzyme shape so the active sites are closed, meaning that it inhibits an enzyme. And so there is cooperativity. And so some substrates are going to bind to one active site on the enzyme and stabilize that active form of the active site. Feedback inhibition. And so sometimes our end product of a pathway kind of acts as an inhibitor to an enzyme earlier in that pathway. And so the end product inhibits that pathway. And so we start off with a substrate binding to our enzyme. Okay, an intermediate molecule is going to be made and moved to another enzyme. It's going to pop off and produce a product, which then that product is the correct shape to bind to that allosteric site on enzyme 1, inhibiting this metabolic pathway to occur. Remember, it changes that shape, and now that original substrate can no longer fit in enzyme 1. And so it'll go until all of those enzymes that were producing this product are now inhibited or changed. All right, that's it for this section.